It's my pleasure to now introduce our second speaker. This is my colleague, Dr. Martin Wuhr. And Martin is assistant professor at Princeton University. He's in the Department of Molecular Biology and the Louis Sigler Institute for Integrative Genomics. And Martin, I really love on your website, you have the statement that said, how do molecules organize into living systems? And I thought that was really quite interesting for, a slightly more analytical professor to have that statement. So I love to hear what that actually means, Martin. Thanks so much.
Thank you so much for the opportunity to talk here. I mean, this might be a way to actually admit that I'm not by training an analytical chemist, but actually started out as a biochemist and only got towards mass spectrometry as a postdoc. But so I'm generally just very much interested in how proteins organize. One second.
Martin, let's put into the slideshow.
Yes, can you see this?
This is the preview. Yep, perfect.
Now it works? Okay.
Perfect.
Wonderful. Okay, and so one of the questions that my lab is interested in is how protein abundance is controlled, which is one of the key organization principles of a cell. And obviously, we all know that there's various levels on how protein abundances can be changed. So we can have more or less transcription, more or less translation, and then also modulate the stability of a protein and therefore change abundances up or down. One second. So with standard genomic approaches like RNA-seq or ribosomal profiling, we have actually pretty good access to transcription and translation regimes. But measuring protein degradation is still probably a little bit lagging. And so there's one method which is very straightforward to measure protein degradation, which uses inhibitors of translation rates, like, for example, chloramphenicol in bacteria or cyclohexamide in a eukaryotic system. And so here the basic assumption is that you do not change the system at all. You inhibit the production of new proteins. And so now if we follow the level of a protein with Western blots or with mass spectrometry, you just immediately can read out the degradation rates of all these proteins. So this is fairly widely used. And so I wanted to study protein degradation, or we wanted to integrate all these different levels of regulation for protein abundance. And so we want to do this in bacteria. And this was one of the very, very first things I did in my lab when I started 6 years ago. And we did this in bacteria, and then with mass spectrometry after drug addition, followed the protein levels. And we see this nice, most proteins are stable, and then there's fewer and fewer proteins which decay faster and faster. So this is very, very straightforward until we started to look more into the data, and we became more and more suspicious because somehow all the periplasmic proteins, for example, were decaying fairly rapidly, and suspiciously many of them were decaying. Until we started to look a little bit more into what might be going on, and we found out that these periplasmic proteins are actually not degraded at all, but somehow upon it, and only upon addition of these translation inhibitors, and we can do this with various translation inhibitors, the periplasmic proteins are actually secreted into the media, into the supernatant, and because we only collected the cells after spin down, we actually thought that the proteins are degrading while they're actually not degrading, but getting kicked out. And we can confirm this by quantifying protein abundances in the cell and or the supernatant. So this was quite a surprise for me, and kind of suggests that these standard translation inhibitors can have some major side effects, and one has to probably be very careful on interpreting the results for protein stability studies. So instead of perturbing the cells very heavily, we went back to a very classical way of measuring protein turnover by using isotope tracing. So we are still in E. coli, and to make analysis a little bit simpler, we go into a chemostat to enforce steady state, and then we switch the feed of the media from light isotopes to heavy isotopes. And obviously, over time, we now can follow more and more heavy isotopes will be incorporated into the proteins of these E. coli cells. So if one would now acquire spectra at various times after heavy isotope addition, one would expect to see the isotopic envelopes of these peptides to shift more and more to the right. And obviously, if I give you any of these kind of measurements, you can fairly easily calculate back what is the turnover time for these kind of peptides. So in theory, this is all very easy. In metabolomics, people do this obviously all the time. But it turns out if you apply the same kind of approaches for proteomics, the system is actually a lot more complicated, and it's quite difficult to make these measurements and to interpret these measurements. So here are some actual spectra acquired, or MS1 spectra for a given peptide. The top spectra here is before the label switch, and before we add the heavy isotopes, there's many peaks, but for many isotopic envelopes, you still can kind of do a decent job of assigning the peaks to the corresponding isotopes. However, once we add the ammonia or the heavy ammonia, now all the isotopic envelopes of these peptides are heavily spreading out and kind of are growing into each other, and becomes nearly impossible to confidently assign peaks here to the corresponding spectra. And so quantification at this level, or turnover quantification at this level, becomes very, very difficult, except perhaps for the highest abundant peptides and proteins. So to overcome this problem, we made things even more complex. We combined this heavy isotope labeling with isotope tracing experiments, where we now label each of these conditions with an isobaric tag, in this case TMT Pro, combine them all together, and then combine them all together, and then we program the instrument to preferably go after the pseudo monoisotopic peak. So when we then quantify, we are using a so-called complementary Porter ion strategy. So we are not reading out this low MMOC part, which mostly is read out for multiplex proteomics, but we are reading out the balancing part of the peptide or of the isobaric tags together with the peptide. And this strategy has the unique advantage that complementary Porter ions from different peptides have slightly different masses. And so therefore, with a high resolution mass analyzer like an Orbitrap, we typically can distinguish what is real signal from what is background noise. And this is extremely important for these kind of spectra, which are so extremely complex. And so we can do very accurate measurements at the MS2 level and measure the true contributions of this peptide that we are really interested in. Here's some actual data for two example proteins. So OMP-F is a protein in the membrane, which our friends who know E. coli well tell us this should be very stable. And each of these dots here is one peptide at one time measured for this OMP-F protein. And so this is all still in the chemostat. And we have here a theoretical dilution limit in the chemostat, simply set by how much of the medium is coming in and how much, which has to be the same as flowing out. And so this way we set the cell doubling time. And we set this here to 6 hours. And the dotted line here, that's our fitted protein turnover, which is a little bit longer than this theoretical limit, but it's a fairly close match on how fast these proteins are turning over. So here we would expect to only see the replacement rate of the proteins. And so this is the theoretical slowest turnover rate these proteins could have. In contrast, RPOS, that's a sigma factor. This is expected to turn over a lot more rapidly. And indeed, when we do here the fit for these proteins, sorry, for the corresponding peptide, we indeed see that it has a predicted much shorter protein half-life. So obviously I can always find two proteins that look good when I measure thousands of them, but that this is actually overall working really well is exemplified here by the scatter plot, where we did two complete replicates, two different people in the lab, 1 year apart, for all these proteins here. So for each of these graphs on the left, we can get the half-life of these proteins, and each dot here represents these two half-lives from the plots here on the left, and overall we see pretty good agreement, and RPOS and OMP-F are not particularly unusual good measurements. So with this method at hand to measure protein turnover in E. coli cells on a global scale, we can now ask, how does protein turnover change under various different growth conditions? And the first growth condition we changed was cell doubling time. So because we are in the chemostat, we very easily can adapt cell doubling time by simply changing the inflow of new media into the chemostat. And obviously the overall replacement rate is a combination of active degradation plus the dilution coming from cell division. And one can come up with two very, very simple models. One would be that this active degradation scales with dilution. So this way, everything would be nicely constant. All protein levels would be unaltered, regardless of how rapidly cells divide. And an extreme other model is that this active degradation rate is completely uncoupled from the cell division rates. And so this would mean that protein levels have to change depending on how fast these cells are growing. We cannot directly measure the active degradation rates. We only can measure like the total replacement rate, which is the combination of the two. But importantly, these two models have very different predictions when we now compare the T-half of cells of all the proteins measured under different division rates. And so if model 1 would be true, then everything should be scaled. And so everything should just be here on this 1-to-1 line. If active degradation rate is uncoupled, we should get here this curved line. And so we can now go in and just do these measurements at various doubling times. And when we do the comparison with the theoretical and compare here the measurements with our this very simplified two models, we see that regardless which condition we compare and what doubling times we compare, we always have the model 2 heavily winning, which is the model where active degradation rate is completely uncoupled from the cell division rate. And obviously, biology has to be more complicated. Some of these proteins down here clearly tend to follow more the other proteins, the other model, and some of them seem to make sense. These might be guys where the cell has some interest of actually modifying active degradation rate. But overall, I believe this is very convincing that and kind of surprising that the typical protein in E. coli very clearly follows this one very simple model. And so typically it seems that the active degradation rates of proteins are mostly uncoupled from the cell division rates. So another another question we could ask is, if we keep now cell division rates constant, but change nutrient limitation? How does cell division rate, how do degradation rates change? And so everything I've shown you so far was done in carbon limited cells. This is now a slightly different representation where I show a histogram of these T-halves. We are now only comparing cells which divide every 6 hours. So this here is our theoretical limit. And most proteins here show a half-life which is consistent with the proteins being perfectly stable and only being replaced due to cell division. Here on the right side of the plot, this has to be due to measurement noise. And so if you mirror this, this gives you probably some sense of the noise in the system. And so this is, when you do it a bit more complicated, we developed a noise model. And so here, indeed, we find that about 20% or so of the proteome is confidently actively degrading, while the vast majority, about 80% of the proteome is very, very stable. And there's a fairly small subsection of proteins which are turning over really rapidly compared to the cell division rates. If we now do the same kind of measurements, but go into phosphorus limitation rather than carbon limitations, we see very similar results. And it turns out that most proteins which are turning over rapidly here are also turning over rapidly here. And we have, again, about 20% of the proteome confidently degrading. However, when we go to nitrogen-limited conditions, the entire histogram changes fairly drastically. And we now seem to see that a large fraction of completely stable proteins is now starting to become slightly unstable. So a large fraction here is starting to turn over slowly. And also here, the fraction of the proteome where we are very confident that they are turning over increases here from 20% to 50%. And this is probably still a heavy underestimation of how many proteins are here turning over, because here many of these proteins are just very close to the stable state, and due to measurement noise, we cannot make this confident call. So we were quite surprised by how many proteins here are stable, how many proteins here became unstable, and we're quite puzzled by these results until we started to partition the proteins into their subcellular localization. And so this is the same graphs that I just showed you, but here the proteins are labeled based on their subcellular localization, cytoplasm, periplasm, or membrane protein. And everything looks pretty much the same under all three conditions for periplasmic proteins and membrane proteins. So there, the mode for all these is at the expected cell division rate of around 6 hours. But the cytoplasmic proteins are quite different under these three different conditions. In carbon and phosphorus limited, also most cytoplasmic proteins are essentially perfectly stable. But under nitrogen limited conditions, it seems that most cytoplasmic proteins are now actually at least slowly degraded. And there's essentially no cytoplasmic proteins which is perfectly stable anymore. And so this makes probably a lot of sense. So if you're running out of nitrogen, but you still want to have the ability of making new nitrogen, then you might be willing to, if you are running out of nitrogen, but you want to make new proteins, you might be willing to sacrifice some old ones so that you have a permanent influx of new released nitrogen or new released amino acids to be able to respond to outside stimulus. And if we do some back of the envelope calculations, it actually turns out that about 2/3 or so of the cellular nitrogen storage is in proteins. And I just wanted to show the same data that I just showed you in a slightly different form here. So this is in scatter plot, where we have the nitrogen limited half-lives versus the carbon limited half-lives, both here for periplasmic and membrane proteins. Everything here is centered with contour plots on the perfectly stable on the 6-hour condition. But here you see this very clear shift of the cytoplasmic proteins from stable in carbon limited to slowly getting degraded in nitrogen limited. And we could also show that this is not just a chemostat kind of interestingness, but this also applies to standard batch culture grown bacteria. So here, when we grow up E. coli in batch culture and then switch them over to nitrogen limited medium, we indeed see here that, again, the cytoplasmic proteins after this switch are slowly degrading, while membrane proteins are stable. And if we do this here for chemostat or for batch culture, we get fairly similar results. A very obvious question is, what are actually the underlying protease responsible for this slow turnover? And so in E. coli, there's 4 ATP-dependent proteases known, and we expect something here to be ATP-dependent because many of these proteins have very clearly defined structures, and it just costs energy to unfold these proteins and then make them amenable for proteases. And so the 2 famous ones are CLIP-P and LON. And then there's more exotic ones, HSLV, which is believed to have very few substrates, and FTSH, which also is believed to have few substrates. And then additionally, because one of the substrates is very key, its knockout is unfortunately lethal. So in E. coli, it's very simple to generate knockout strains, and we now repeated these measurements in a strain where we knocked out CLIP-P with the wild type. And so if nothing changes, we should be here on this 1-to-1 line. And if a protein gets stabilized upon knockout of this protease, we expect here a shift towards the right. And so CLIP-P here, we see a little bit of a shift towards the right. And then with replicates and noise models, we can label here in purple the proteins which we are confident to be degraded indeed by CLIP-P. And it's a surprisingly small fraction of the entire proteome that gets stabilized with CLIP-P. There's still a lot of degradation happening here once this canonical protease is knocked out. Okay, what about, and we see essentially no shift in terms of the cytoplasmic stability of these proteins. So we would expect this to entirely move over here. The other canonical ATP-dependent protease, LON, also doesn't look a lot better here. HSLV is, we essentially do not detect any protein which we confidently see to be degraded with HSLV. And so we were wondering, is it possible that these proteins are just, or these proteases just act in a redundant manner? But so in E. coli, we can very easily combine all 3 knockouts. And we see here more degradation stabilization than even if we just sum up the stabilization in the individual knockouts. But overall, it's really surprising how much degradation is still happening here, despite here the 2 canonical and then even the 1 more exotic ones being knocked out. Obviously, here we couldn't add the knockout of this lethal protease FTSH. Our friends in the Silhavi lab were able to actually knock out FTSH by also modifying the downstream enzyme that makes the lockout of FTSH lethal. However, these cells are really sick, so we cannot grow them in the chemostat. They're just getting diluted out. But when we go back to our batch culture experiments, even in this FTSH knockout here, we see very clearly that the cytoplasmic proteins are degraded upon nitrogen condition. So together, it seems that even though E. coli is this best studied of all model systems, probably we seem to be missing a major proteolytic pathway. So together, none of these proteases seem to explain this major turnover. And even here in this triple knockout, we have more than 50% of the overall proteome turnover still happening. So I think this is very interesting. What we are still missing here, and the lab is now very heavily involved in trying to follow up on what is this protease or this missing mechanism that is still to be discovered. But so I started the talk with we try to understand global protein abundance control, and I motivated with can we now bring everything here together? And it's obviously very simple that protein levels correspond to transcription rates, translation rates divided by degradation rates. We can now we can get all these measurements now, RNA-seq, ribosomal profiling, and then degradation rates of what I just showed you. And we have to modify things a little bit because, as you all know, we are not very good with measuring absolute protein abundances with proteomics. So we go into relative abundance space so that we can use standard isotope labeling. And so now we can here ask how much does RNA-seq fold change if we compare two different conditions, explain the observed protein abundance change. And it's the typical ask where of around 40% that we see. If we add only the translation rates obtained with ribosomal profiling, we add another 8%. And if we add in the degradation rates, this is a separate 8%. But what becomes now really exciting is if we can integrate everything together, we can explain about 70% of the observed protein abundance change based on these three measurements. And what's, I think, quite exciting about this, even though there's still quite a bit of missing, is that probably most of the remaining discrepancies are due to measurement noise, predominantly here, probably in the ribosomal profiling measurements. We just have to get better in this kind of technique via mass spec lab, and there's something we have to still learn. So to summarize, I hope I've shown you that we can measure unperturbed protein turnover by combining complementary porter ion measurements on the MS2 level with heavy isotope labeling. We find that active degradation rates in E. coli cells is uncoupled from cell division rates. We see that nitrogen-starved E. coli recycles its cytoplasmic proteins. And lastly, that we can integrate these turnover measurements with other omics measurements to get to the point where we can really parameterize protein abundance control on every single level. And with that, thanks a lot for your attention, and I would be very happy to take your questions.
Sorry, I was muted. Thank you so much, Martin. That was wonderful and super interesting. I wanted to encourage everybody from the audience to please use question and answer feature here in the Zoom. Maybe I'll start with a question. So Martin, I can imagine with this quite complex labeling of the nitrogen, but then also like the reporter ions. So I assume you, I mean, of course, probably would have to write your own software. Is that available for other people to use then also?
Yes, so the software is available on GitHub. Yeah.
Mm-hmm. Okay, that's great. Yeah, okay, good. And then actually the other question I had, have you tried this approach, or are you planning to also look at other organisms other than E. coli?
Yes, so we've used it also for yeast. That's exactly the same method. There's nothing different. We are using it right now for mouse and also for embryos. So my lab does a lot of work with frog and fly embryos. There, the labeling strategy has to be different. So in mice, we can either go with amino acids and or heavy water labeling. In embryos, we have to go with heavy water labeling. And it turns out the main source of proteins made in embryos is old proteins being degraded. So in these embryos, we switch to we essentially take the embryo and put it in O18 water. The peptides are getting cleaved, and then you have the O18 gets incorporated into the carboxy group, and then if the amino acid is recycled, you have a 50-50 chance that the heavy O18 gets incorporated into your newly synthesized proteins, and it works beautifully in this system. So. Yeah,
very elegant. I think Jeff has a question also.
Very cool talk, Martin. Thank you. I'm wondering, do E. coli secrete proteins into the media? Do they release them per se?
Yes, that's a that's a very interesting question. So because we were so scared with the results that we had with chloramphenicol, when we collect our samples, we actually take all the supernatant and just evaporate the water so that we don't have to distinguish between what is the proteins that are in the supernatant versus what's happening internally, so that we don't have to separate this out. But I think so. So at the moment we don't know. But I think it's a very interesting question of how much of what might be going on might be explained by this. Yeah, how much of it is due to separation or so.
Yeah, it could be minor, could be substantial, hard to say. That's very interesting though. Thank you.
Yep.
Well, thank you, Martin. This was amazing. And I wanted to also highlight that Martin, I like to say the German way, Martin actually was recipient of the 2021 Young Investigator Award. So, you know, it's great to see you a few years later here again, still involved with US HUPO. So I love that. And yeah, thank you very much for your talk. I really enjoyed this. Thanks, Martin.
Thank you.
Okay, great. Well, this brings me to our next speaker.